continuing on here in chapter 15, talking about some more of those second line defenses like phagocytosis and inflammation in the complement system. Now moving into some of our more second line defenses, so phagocytosis, so the kind of general activity is, again, their surveillance. So they're looking for things, but if they discover a microbe or something that doesn't belong or just a dead cell, essentially phagocytosis is just ingesting or eating that material. And the goal is then to eliminate it. So it recognizes something, something that doesn't belong. So some type of foreign matter on it. And then those phagocytes, again, ingest it and get rid of that material. And there are three main phagocytes in our body. So neutrophils, monocytes, and macrophages. Again, not all cells go through phagocytosis. These are the three main ones that can. Now, the stages are kind of overall process of phagocytosis. The first thing is chemotaxis. And so we think about taxis, moving thing, and chemo for chemicals. So this is just going to be the movement of a phagocyte to an area. So again, if there's a foreign invader, chemicals get released and it attracts these phagocytes. So the actual movement, kind of intentional movement of the phagocyte to the invader is called chemotaxis. So it's being moved intentionally. Once it attaches to it, so it has to attach to something that doesn't belong, then it's actually going to ingest. So we start to see that this phagocyte starts forming what we call a vesicle, and it engulfs that material. So it's bringing it in. Then it joins that with those lysosomes. So remember, lysosomes are kind of the trash cans of the cell. So the lysosomes start fusing and joining in to that, and they release all of these enzymes that are used for digestion, and ultimately it destroys it. So it breaks down the bacteria or the substance into small little pieces, and then ultimately the cell gets rid of it by exocytosis. So it just releases the leftover contents, the broken down material, out of the cell. So that chemotaxis and ingestion, we come back to, again, how does the phagocyte know what's foreign and what isn't? So it's those PAMPs, those pathogen-associated patterns. Those are recognized by the phagocyte. And again, it serves in as a signaling molecule that's there on the surface. Now, these are not present in mammals, so we don't produce these. And this is, again, a way of training the body. So they should not be recognizing self cells because mammalian cells don't produce these PAMPs. And just some examples I mentioned before, but again, peptidoglycan, we don't make that. We don't have a cell wall. Lipopolysaccharide, that's only in those gram-negative cell walls. Double-stranded RNA, that's in viruses. That's a way to recognize them. All of our RNA is single-stranded. So if our cells recognize double-stranded, it indicates there's a virus present. Now those pattern recognition receptors, that's what's receiving or recognizing. So those are going to be on the surface of the phagocytes or other immune cells like our lymphocytes or dendritic cells, and they recognize and bind to those pathogen-associated molecular patterns, and that is going to activate them. So it causes phagocytosis, or it causes a reaction meant to get rid of them. All right, now moving into inflammation, another second-line defense. So we've all, I'm sure, undergone inflammation. So this is very common, not just in an immune reaction, but any traumatic event. So we think about like a broken ankle or something like that that can lead into this. So it's very easy to identify using kind of this set of characteristics or symptoms. And these are from Latin terms, so they're a little unique. So rubor, so that's redness, so that kind of goes along together. Calor is going to be the heat, and that's as blood is coming in for that chemotaxis, as we're bringing the immune cells to it. We get some heat from the extra blood. The tumor, which we think about, of course, cancer, but here we're talking about swelling, so we're getting lots of edema. Again, that's where that lymphatic system helps drain that extra fluid once inflammation's over. Dolor is the pain that gets associated. So all of those are from those Latin terms. That's why they're kind of weird. But then also the fifth symptom of inflammation is a loss of function. So again, it's not able to move. We can't bend if we have a swollen or an inflamed ankle or wrist. It just makes it really hard to use it. Now, why is this part of our immune system? So what are the functions? So it's a few things. First of all, it mobilizes and attracts other immune cells. So it helps bring the B and the T cells for the acquired immunity to the area that has the trauma or the problem. So it sets in repair mechanisms to get, once that inflammatory response and once the immune response is over, it helps repair everything and put it back to normal. And then it also helps to destroy the microbes in blocking further invasion. So it helps localize the infection. So if you get a cut and it gets inflamed, it's one way of trying to keep the microbes from getting into the blood and then moving throughout the body. So then again, just looking here at those major events. So we have kind of our injury here, that immediate reaction. So we have a nail or something going into our skin. The first thing is going to be the vascular reaction. So the vasodilation, the blood vessels get water. We bring lots of blood there. That's when the redness and the heat's going to come in from that extra blood. But that brings those immune cells to the area. Then we're going to get the swelling and the pus. So as extra fluid is being there, the pus is coming as those neutrophils and as those macrophages are chopping up and breaking down through phagocytosis and releasing all the extra stuff. That pus gets formed. And then finally we get the actual repair. So we're going to put everything back to normal, but it can lead, in some cases, depending on the injury, to that scar formation. A little bit more here about the edema. Again, that's the leakage of those fluids. So these are vasoactive substances are going to cause those endothelial cells to contract and form gaps. So this is going to do a couple things. It leads to exudate and to edema. So that exudate is the blood-borne components that escape into that extracellular fluid. And the contents vary with different plasma proteins that are there, like clotting factors or blood cells. And that can be clear, or it can be that pus that has those red blood cells or some of the neutrophils left over as they're going through phagocytosis. And then the other part here is that edema, that's the swelling and the firmness, and that's just due to everything accumulating from that exudate. So again, our lymphatic system, though, during the repair helps drain and get rid of all that extra. Now, a really unique characteristic here is this diapedesis. And this is where those white blood cells, so here we have a white blood cell, like a neutrophil that can go through phagocytosis. Again, if we think about phagocytosis, we need chemotaxis. It needs to get to the area where the invader is, but then it's carrying here in the blood. Well, how does it get from the blood out to the infection? Well, it literally squeezes in between the endothelial or the red blood cell lining. So the squeezing of these cells through the blood vessels out into the surrounding tissue is called diapedesis. And you can see it here under the electron micrograph. It's showing this red blood cell or this white blood cell literally squeezing out of those blood vessels. So this is referred to as diapedesis. It's how the cells of the immune cells get from the circulatory system out into our tissues. So the benefits then of this is that influx of fluid helps to dilute any toxins that are being released from the microbe or from the pathogen. The fibrin's there to clot it. That's going to trap the microbe, again, keeps it from getting into the blood system and spreading to other places. Those neutrophils that go through phagocytosis, it gets them there so that they can destroy the bacteria and any leftover stuff that's there. And again, that leads to that pus formation when those neutrophils are present that accumulates in that area. All right, now another one here in our second line defenses is the fever. So fever is just an adjunct to inflammation. So they go kind of hand in hand. So inflammation and fever typically coexist. So the fever here is just an abnormally high body temperature that is intentional. It's a universal symptom that there's some type of infection going on. It's one of the first things that they do whenever you get to the doctor, or one of the first things they'll ask. Now, it can also be associated with other things. It doesn't just mean it's an infection. It could be allergy, even cancer, or other illnesses can lead to a fever. So it's not always an infection or pathogenic problem. And then just to put it into terms here, and we're mainly familiar with these, so anywhere between 100 to 101 is considered low-grade fevers unless you're an infant, and then we start caring more about those lower grade. And typically that indicates a viral infection. Then moderate is 102 to 103. And then 104 to 106 is very high. These are more indicative of bacterial infections, but especially a high fever. So fevers are good. They're part of our immune system. They help activate our immune system. They make it harder for microbes to replicate, but they can get too high to where our proteins can start to denature and we start to get problems. So prolonged high fevers definitely is something that would need treated. So pyrogens is the term here that we use. This is actually a chemical that causes a fever. So again, it's intentionally telling the hypothalamus to increase the thermostat and make it hotter to cause that fever. There are exogenous pyrogens, as the name implies. They come from outside of the body. So things like viruses or bacteria, any type of invader, endotoxin, things coming from outside that cause a fever. Those are exogenous pyrogens. Endogenous pyrogens, those actually come from inside of the body. So this goes along with inflammation. So again, when inflammation occurs and we get activation of monocytes and neutrophils, that also triggers a fever. But those chemicals are coming from inside the body, so they're endogenous. And then also a few other chemicals like interleukin-1 and tumor necrosis factor can also cause fever formation. Now, why do we want to keep that fever? Again, typically one of the first things we do is pop in an ibuprofen or something to bring the fever down because we feel miserable. However, it's beneficial. It inhibits multiplication of sensitive microbes, so they can't reproduce, at least not as quickly. It gives our immune system time to mount that defense. It also impedes the nutrition by reducing availability of iron. So a lot of microbes need iron. Whenever the fever goes up, that gets imbalanced, so it slows down reactions that are needed for growth. Their enzymes don't work well at a higher temperature, but ours do. So it increases our metabolism, increases and stimulates our immune system, but inhibits bacterial growth. Now, the treatment of fever, again, difficult decision. Again, so there are some advocates that even a slight to moderate fever in otherwise healthy person should be allowed. Just let it run its course. It helps get rid of that. So there are potential benefits and minimal side effects. Now, all medical experts, though, agree that high prolonged fevers are extremely risky and they should always be treated. Again, lower grades, again, if somebody's healthy, they can go untreated. Again, that's each physician, each healthcare is going to have their own kind of policy as far as all that goes. But anyways, high fevers though, or fevers for a long time, that's whenever we start causing problems like cardiovascular disease, can lead to head trauma or even seizures. So again, all the time, those would be treated. Now, some antimicrobial products that can get produced by that second line defense, interferon. This is where we start talking about those viruses. So this is a small protein that's made by some of our white blood cells and some of our tissue cells, but it's used actually in therapy against viral infections. So it's, kind of something the body makes, but it can also be a treatment. So during viral infections, interferon gets made, but it can actually be given as a treatment as well. So it's involved again against those viruses and some other microbes as well, but it just helps regulate the immune system and it helps with communication. Now, what does this antiviral interferon look like? It is not virus specific, so it's not like it just recognizes HIV or it just recognizes like the herpes virus. It's just the synthesis and response to one cell type that protects against other cell types. So it's produced industrially, again, though, as a treatment. So again, our body naturally makes it, but it is also a drug that can be given, made in a pharmacy and prescribed as well. But essentially, what the interferon does is it alters. So the interferon attaches here to specific receptors, and it prevents viruses from assembling. So they may come in. They may still penetrate, but they are not going to go through this assembly process that normally would happen. Another antimicrobial product we'll talk about is the complement system. So this is actually about 50 different proteins that make up, that's why it's called the complement. They all work together to destroy bacteria, and it leads to this cascade reaction. So it's not just like one event, it's multiple kind of sequential responses. So that first substance in the chemical series activates the next, and then so on until we get to that end product. Now, there are 3 complement pathways that we'll talk about: the classical, the lectin, and the alternative. Now, alternative doesn't mean it's like the second option. It's really just another way. So it's kind of misleading in the naming there, but it's not like classical is like the main way. Again, these are just kind of how they're named. Fortunately, doesn't always help us with their function. But the classical complement pathway, they're all different in how they get activated. So the classical pathway is activated actually by antibodies that are bound to a surface. So whenever antibodies are made by the acquired immune system, these complement proteins can recognize the antibody and it gets rid of whatever it's attached to. The lectin pathways, the name does imply here, is activated by lectins that bind to sugars on those microbial surfaces. So again, this is something unique to microbes that we don't have. So there are complement proteins that recognize those lectins. And then the alternative pathway, this is activated by repeating molecules on the surface. So kind of, again, like those PAMPs. So things like the lipopolysaccharide that's on gram-negative bacteria. So it's something foreign. It's not lectin, but it's something foreign that those complement proteins would recognize. And then what happens then is different. So whether we get, again, just the presence of antibodies here attached to something and our classical pathway gets activated, whether it's lectin on the surface of a microbe or whether it's some other molecule that's foreign like the lipopolysaccharide, regardless of which pathway gets activated, we can have a few different things happen. So we get one complement protein that activates another protein that activates another one that activates another one, and so on. That's the cascading event. Well, there are 3 ultimate end goals that can happen with complement activation. So one actually triggers that phagocytosis we've already talked about. Some of the complement proteins can form these membrane attack complexes that literally put a bunch of proteins in the cell wall or on the surface of microbes to destroy them. And then the other one is we can also get the triggering of inflammation. So again, all part of those second line defenses, right? So we'll come back then in chapter 16 and learn more about that specific acquired immunity.